Your unknown mixture will be provided to you in a vial. The first thing you should do is write down your unknown number in your lab notebook. Next, obtain a 100 milliliter beaker and measure its mass. Record this mass in your lab notebook. You're going to need it later. You may notice the last digit on your scale fluctuating a little bit. That's okay, just write down the number that it seems to settle on most often. Now, we want the mass of our solid unknown, so I'm going to tear the scale and then transfer the entire contents of my vial directly into this beaker. The mass that's displayed on the scale will now be the mass of my unknown mixture. I should record this mass in my lab notebook as the mass of the unknown mixture. Recall that your unknown mixture contains 3 different substances, each with different physical and chemical properties. To separate out the sodium chloride, we're going to extract it with water. Sodium chloride is soluble in water, the other substances are not. Use a graduated cylinder to measure 25 milliliters of deionized water. We use deionized water for scientific experiments because it doesn't contain any of the ions that are present in tap water. Pour the deionized water directly into your mixture of the 3 substances. Swirl the mixture for about a minute to make sure that you've fully dissolved all of the sodium chloride that's present in your mixture. Set your mixture aside while we prepare the next setup. Obtain a 600 milliliter beaker and a large stir bar. We want the mass of both of these together. Zero the scale and place the 600 milliliter beaker with your stir bar on the scale to record the mass. You're going to record this mass in your lab notebook as the mass of the 600 milliliter beaker plus the stir bar. This beaker is what we're going to use to collect your sodium chloride solution in the gravity filtration that we'll set up next. To set up your gravity filtration, you will need a ring stand, a ring, a filter funnel, and a large piece of filter paper. Attach the ring to the ring stand and then place the funnel inside of this ring. We're going to collect the solution in your 600 milliliter beaker, so that will be placed underneath your funnel. We need our piece of filter paper to fit inside of our funnel, so we need to fold it into a cone shape. To do this, fold it in half and then fold it in half again to mark the center point of your piece of filter paper, and then take one of those folds and tuck it inside one of the others. This takes a little bit of practice and you'll need to adjust it to make sure that it fits snugly within your funnel. Place your cone-shaped filter paper into your funnel and then adjust it a little bit more to make sure that it fits appropriately within the funnel. It's going to pop out a little bit, so we're going to get it wet to make sure that it stays seated within the funnel. Use a DI water bottle to squirt some water onto the filter paper to get it damp so that it stays seated well in the funnel. You can let this water run directly into your 600 milliliter beaker, and then you can discard it in the sink. Your mixture has now had time to settle so that the solids are toward the bottom of your beaker and the solution is at the top. We're going to decant this into your filter funnel. What that means is when you pour this solution into your filter funnel, you want to leave behind the solid. We don't want to transfer that solid into the filter funnel because then we'll just have to wash it back out later. So you're going to pour that into your filter funnel, being careful to leave the solid behind. Next, to make sure that we've gotten all of the sodium chloride dissolved from our mixture, we're going to do this wash again. So you're going to measure 25 milliliters of deionized water into your graduated cylinder, and then pour the water into the mixture of solids contained within your beaker. Swirl the water around for a few seconds to dissolve any remaining sodium chloride, and then let this sit and settle while your water is draining through your filter funnel. Once it's had time to settle and you can see a clear separation between the solid and the solution in your beaker, you're going to again decant the solution into your filter funnel, leaving the solid behind in your beaker. It's okay if you have a little bit of water left behind with your solid after decanting. that won't affect our results here. You're going to repeat the washing process for a third and final time, again measuring out 25 milliliters of deionized water, swirling your solution, and then setting it aside to settle. You want to let it settle so that we can decant the solution, leaving behind the solid again for a third time. This process does require some patience. Gravity filtration takes some time, so you should expect the 3 washes to take you approximately 15 minutes to complete in total. After your solution has settled for the third time, you're going to decant the solution off of the top, leaving behind the solids in your beaker. At this point, we've successfully separated the sodium chloride from the silicon dioxide and the calcium carbonate. The solid silicon dioxide and calcium carbonate are left behind in your beaker, while the sodium chloride is dissolved in the water in your 600 milliliter beaker. Our next step is going to be to remove the water from the sodium chloride by heating it until it evaporates. Once all of the water has run through your filter funnel and your filter funnel is dry, you can take the 600 milliliter beaker containing your sodium chloride and water solution and place it on a stirrer hot plate. Set your other beaker below the funnel in case there are any residual drips. Turn your hot plate to a temperature of 200 degrees Celsius so that we have plenty of heat to evaporate the water. We are also going to turn on the stir plate. The stir plate agitates the magnetic stir bar that we have in our beaker so that we can get the water to evaporate more quickly. Set this aside as it will take approximately 1 hour for your water to fully evaporate. While your water is evaporating, we're going to carry on with the next steps of the procedure. If you have any residual solid in your filter paper, that's okay. We're just going to rinse that back into our beaker so that we have all of the solid collected together. To do this, you're going to use a bottle of DI water and squirt small amounts of water while you tilt the funnel over your beaker, and you should see that solid rinse into your beaker. You can do this a couple of times, but try to avoid using too much water at this point. Next, bring a small clean beaker over to the fume hood and get a small amount of hydrochloric acid. You should fill your beaker approximately up to the 10 milliliter mark. Back at your lab bench, use a graduated cylinder to measure out 8 milliliters of hydrochloric acid. You want to make sure that you get down at eye level so that you can see the meniscus right at that 8 milliliter mark. Pour the 8 milliliters of hydrochloric acid into your mixture of silicon dioxide and calcium carbonate. You'll recall that the hydrochloric acid reacts with the calcium carbonate, but it does not react with the silicon dioxide. The chemical reaction that occurs here converts our insoluble calcium carbonate into the soluble compound calcium chloride. Make a note of your observations as this chemical reaction occurs and swirl the reaction mixture for approximately 1 minute. You want to make sure that all of the fizzing has stopped. As the reaction proceeds, you should start to see that the calcium carbonate, which was formerly a white, very, very cloudy, milky solution, starts to dissolve, and you're going to have a much more clear solution. If it looks like your calcium carbonate is still present in solution, you can add an extra milliliter or 2 of hydrochloric acid to get it to fully react. Don't add more than 10 milliliters total of hydrochloric acid. Make sure that if you are adding extra, you swirl the reaction mixture again and wait for the reaction to go to completion. After this reaction is finished, you should be able to see a clear separation between your solution, which should be mostly clear, and the silicon dioxide, which is left at the bottom of your beaker. Remember, your solution at this point contains dissolved calcium chloride, which we can now separate from the silicon dioxide because we did that chemical reaction. To separate the soluble calcium chloride from the silicon dioxide, we're going to decant the solution. That solution at the top is called the supernatant. We're going to decant the supernatant, leaving behind the precipitate, which is our solid silicon dioxide. We want to wash the solid silicon dioxide with small portions of DI water, so you can squirt just a few milliliters of DI water onto that solid, swirl your beaker to mix it around, and then decant once more. Repeat this washing process a second time to make sure that any residual calcium chloride or hydrochloric acid or any of those other substances is rinsed off, leaving behind just our silicon dioxide. Place your beaker of silicon dioxide into the oven so that we can dry off any of the remaining water. Make sure that your beaker is labeled and that you know which one is yours so that you can identify it later on. At this point, we've successfully separated the three components of our mixture, but we need to recover the calcium carbonate. Right now, what we have is calcium chloride. To recover the calcium carbonate, we're going to react the calcium chloride with sodium carbonate. Measure 15 milliliters of sodium carbonate into a graduated cylinder and then pour that into your solution. This chemical reaction converts the soluble calcium chloride back into the insoluble calcium carbonate. Record your observations as this chemical reaction occurs. If after adding 15 milliliters of sodium carbonate you don't notice a solid precipitate forming, add a little bit more. You can add this in 5 milliliter increments. Measure out an additional 5 milliliters of sodium carbonate and pour that into your reaction mixture. You can repeat this process again if you still don't have any solid forming. Now that we've recovered our calcium carbonate, we need to perform a filtration to collect the solid and separate it from the water and any dissolved salts that are present in solution. To perform the vacuum filtration to collect our calcium carbonate, you will need a filter flask, a hose, a Buchner funnel, a filter adapter, and a piece of filter paper. We're going to collect the solid calcium carbonate onto the filter paper, so we want to make sure that we record the mass of the filter paper in our lab notebooks. Take the filter paper over to the scale and record the mass of the filter paper in your notebook. To set up the vacuum filtration, connect one end of the hose to the vacuum line and the other end of the hose to the sidearm on your filter flask. Place the filter adapter in the mouth of the filter flask and then place the Buchner funnel on top. Lastly, place your piece of filter paper onto the Buchner funnel. Wet the filter paper with a small squirt of deionized water before beginning your filtration. To begin your vacuum filtration, turn the vacuum line on. You do this by turning the handle parallel to the hose. With the vacuum on, slowly pour your solution over the filter paper. You want to pour slowly so that you don't accidentally knock the filter paper off of the Buchner funnel. Rinse any residual solid left behind in the beaker using small portions of deionized water. You now have your solid calcium carbonate collected on the filter paper. Leave the calcium carbonate on the filter paper with the vacuum on for about a minute to let it get a little bit dry. After about a minute, turn the vacuum line off and detach the hose from the sidearm of your filter flask to break the vacuum seal. You should then be able to easily lift the Buchner funnel off of the top of your flask. The filter paper is still a little bit wet, so we want to put it in the oven to dry. Carefully scrape the filter paper off of the Buchner funnel, making sure to keep all of the solid contained on the filter paper. Transfer this to a watch glass, which we can then place in the oven. Write your initials on your watch glass so you'll be able to identify your sample, then place your sample in the oven for at least 10 minutes. This will give it time to fully dry. While waiting for your calcium carbonate to dry, you can remove your beaker of silicon dioxide from the oven. Do this using an oven mitt since the glassware will be hot. Set the hot glassware on the countertop to cool before you obtain its mass. Weigh the solid silicon dioxide in the beaker and record the mass in your lab notebook as the mass of silicon dioxide plus the beaker. Recall that you recorded the mass of this beaker early on in the lab, so you can subtract the mass of the beaker from what you just weighed to obtain the mass of just silicon dioxide. After about 10 minutes of drying, you can remove your calcium carbonate from the oven. Again, use an oven mitt to do this since the glassware will be hot. Set the hot watch glass on the countertop and let it cool for a minute before obtaining its mass. Weigh the filter paper, which now contains your solid calcium carbonate. Record this in your lab notebook as the mass of calcium carbonate plus the filter paper. Recall that you recorded the mass of your filter paper earlier on, so you can subtract out the mass of the filter paper to obtain the mass of your calcium carbonate. By this point, your beaker of sodium chloride should be fully dry. Fully dry means that there's no water droplets left anywhere on the beaker. Once you're sure the beaker is fully dry, turn off the heat and the stir and remove the beaker from the hot plate. Do this using an oven mitt and let the beaker sit on the countertop to cool before obtaining its mass. Finally, record the mass of your solid sodium chloride along with the beaker and the stir bar. You're going to record this in your lab notebook as the mass of sodium chloride plus the beaker plus the stir bar. Recall that we recorded the mass of the beaker and the stir bar earlier on, so you can subtract out their mass to give you the mass of just the sodium chloride. You should now have the three components of your mixture separated into three separate locations. You have all the data necessary to determine the mass of each substance that was in your original mixture and to determine the mass percent composition of each. Before you discard any of your samples, calculate your mass percents to make sure that they make sense. You want to make sure that if you need to recollect any data, you haven't thrown out your samples. Once you are confident in the data you've collected, you can dispose of your samples. Solid waste should be disposed of in the solid waste container, and liquid waste should be disposed of in the liquid waste container. Use a funnel to transfer your liquid waste and make sure you cap all waste containers after you're done. Wash all glassware used in the experiment and lightly dry with a paper towel, then return the glassware to its original storage location. No glassware should be left on the drying rack.